So colon cancer is the second leading cause of death from cancer in men and women. And based on projections, represents about 10% of new cancer cases in 2022 and 11% of estimated cancer deaths in 2022. So the gold standard for colon cancer detection usually involves relatively extensive and invasive colonoscopy, which requires specialist doctors and medical care specialists. So a more reliable, inexpensive, and portable test would allow for a better point of care colon cancer screening. So previous studies have established that specific urinary metabolites can differentiate between healthy and colorectal cancer patients, and 3 specific urinary metabolites can be used as biomarkers for detecting colon cancer. So those being diacetylspermine, creatinine, and hippuric acid. And to initially develop the sensor system, metabolites need to be conjugated onto nanocarriers, which are agents that can carry a substrate and cause or amplify a change in electrical impedance of the sensor system. So in this study, we used gold nanoparticles and liposomes. And then the next step for developing the sensor would then be to modify the surface of electrodes with metabolite-specific antibodies and permit the competitive binding between conjugated metabolites and free metabolites, which should cause a detectable change in impedance due to the attachment and detachment of the conjugated nanocarriers. So the basis of the biosensor then relies on electrical impedance spectroscopy, and impedance and phase both depend on frequency, and a characteristic impedance curve has 3 distinct areas and a parabolic phase. And the impedance curve can be divided into 3 sections, the first being dominated by double layer capacitance, the second being dominated by the resistance of the solution, and the third being dominated by the geometric capacitance of the electrodes, which can be seen in the equivalent circuit on the top right. And gold nanoparticles have been shown to provide a capacitive change when bound to the surface between the electrodes on that silicon dioxide surface due to the buildup of 2 layers of charge caused by the gold nanoparticle and surrounding solution. And this should result in an increase in impedance and a downward shift in phase, which can be observed in our graphs. And liposomes are expected to yield a similar effect, although not as robustly documented in the research. So the sensor depends on the attachment and detachment then of the conjugated nanocarriers. And the mechanism by which the gold nanoparticles or liposomes are removed is through a competitive binding system, which has 3 main components. The first being the attachment of a metabolite-specific antibody. So in this study, the creatinine, hippuric acid, or diacetylspermine antibody. And then the second component being the attachment of the gold nanoparticle or liposome attached to the metabolite of interest. So in this project, we had 6 conjugates prepared, and those conjugates are able to bind to the antibody via the metabolite attached to its surface. And then finally, the free metabolite of interest, which should have a higher binding affinity to the antibody, is able to displace the conjugated nanoparticle. And the release of this nanoparticle should then cause a decrease in impedance or a reversal of the effect caused by the initial attachment. So then we were tasked with attempting to modify the silicon dioxide surface with the metabolite-specific antibody. So the first step of that would be to apply a oxygen plasma treatment to allow a uniform layer of hydroxyl groups to be added. Then 3-aminopropyl triethylsilane, abbreviated as APTIS, is incubated to add a amine functionality to the surface. And then incubation with succinic anhydride results in a ring opening reaction that results in the formation of amide bond between the terminal amine and succinic acid. And then the exposed carboxyl group of the succinic acid then binds to the amine functionality of the antibody through the amide bond. And EDC and NHS is used in this reaction to increase coupling efficiency and result in a more stable amide bond. And then after the antibody is added, the surface is incubated with the metabolite conjugated to the liposome or the gold nanoparticle to allow attachment to the antibodies. And based on impedance testing with gold nanoparticle conjugated to acetylspermine conjugates, we were able to observe a 900 ohm increase. And then with the liposome acetylspermine conjugates, we were able to observe a average of 190 ohm increase. And then based on the impedance and phase readings from a single electrode, incubated with the gold nanoparticle acetylspermine conjugates, we were able to see this characteristic increase in impedance after the addition of the gold nanoparticle and a downward shift in phase. And we were able to observe similar trends in our creatinine test as well, where we saw after the addition of gold nanoparticle creatinine conjugates, an average of a 220 ohm increase in impedance. And then after the addition of the liposome creatinine conjugates, we were able to observe an average of a 25 ohm decrease in impedance. So perhaps suggesting a more sensitive system using the gold nanoparticle conjugates compared to the liposome creatinine conjugates, which is, again, a similar trend where we see a more sensitive system with the gold nanoparticle conjugates in the acetylspermine test as well. And again, we were able to see this in a single electrode treated with the gold nanoparticle creatinine conjugates, a characteristic increase in impedance and downward shift in phase. We also performed a variety of microscopic techniques on the surface of our modified electrode to ensure surface homogeneity, as well as validating that the conjugates were actually bound to the surface of our electrode. So we used scanning electron microscopy, or SEM, which gave us some information about the microstructure of our surface and homogeneity and morphology of the surface. So in figure A, it is showing the electrode surface with no modification, and B with just modification with aptus, and C with the surface treated with creatine bodies, and then D, the surface treated with creatine bodies, as well as the gold nanoparticle creatinine conjugates. So observing the attachment of those metallic conjugates onto our surface in that last image. And we also performed atomic force microscopy on the surface of our electrodes exposed to the gold nanoparticle attached to all three different metabolites. So in these images showing the an AFM height image after exposure to the creatine antibody, as well as the GMP creatinine, and then also the corresponding size histograms, which shows a greater frequency of around a 20 nanometer attachment or a conjugate onto its surface, which corresponds to the size of our GNP creatinine conjugates as observed in dynamic light scattering, UV visible, and as well as a variety of microscopic techniques. And then we also performed AFM on our gold nanoparticle acetylspermine, as well as gold nanoparticle hippuric acid conjugates, again showing this greater incidence of these 20 nanometer particles, which corresponds to the size of these conjugates, suggesting that we were able to successfully bind these conjugates onto the surface of the electrode. So the next steps then of this research is to continue to optimize and troubleshoot with the sensor, and testing with actual urine samples and spiked metabolite samples, and just ensuring more consistency, and also working towards the broader outcomes of this project, which were to develop an easily accessible and portable system that can detect the presence of biomarkers for colon cancer. And the use of impedance detection, as shown in this project, provides a novel avenue for point-of-care testing for colon cancer, which has become a increasingly pressing issue. So I would like to acknowledge the University of Alberta and the members of the Wishart Lab for their help on this project, as well as the PI of the lab, Dr. David Wishart, and my direct supervisor, Dr. Yagana Kanani, as well as the NMIN network and NSERC. And I would like to just reiterate at the end of this presentation the three key messages, which are that gold nanoparticles and liposomes can be conjugated to small metabolites, and these conjugated nanoparticles can be detected by attaching to the surface of an antibody modified electrode through impedance signaling, and that optimizing the performance of individual components of the sensor may result in many useful advancements for the detection of metabolites, specifically those shown to be related to colon cancer. So I'd like to thank you for listening to my presentation today. Are there any questions?
Well, that gives me an option to ask a question. Irene, I'm wondering if you could speak to why you use liposomes as the nanocarrier for the metabolites.
So based on some initial testing in this project, it was shown that liposomes have a higher biocompatibility and stability compared to gold nanoparticles, which tend to be more prone to aggregation and are slightly less stable. As well, liposomes have not been as well documented in the research in their use in such a impedance-based biosensor, especially one that uses this kind of competitive binding mechanism. Really has not been documented. Research with liposomes and impedance-based biosensors have primarily focused on kind of their dynamic activity, their and as well as kind of drug delivery systems. So a very novel use of them, which I think was quite informative in this project.
Great, thank you. And what are the future steps that would be involved to optimize the performance of the sensor?
So one thing that we would do is use kind of spiked solutions of the metabolites, which we did initially in our testing, but has not been done as extensively as we would have liked to. And then also using urine samples, which is, I guess the end goal is to be able to apply it to urine samples and actually detect higher levels of these metabolites in patients that are likely to have colon cancer.